Big Imaging, Lecture 8, Part 2, Statistics. This lecture is recorded and will replace the lecture on April 18th. Statistical analysis of experiments. Most of experiments we do in science are usually application-specific, noisy, and often very complicated with many variables. And they are usually not very good as teaching example. So let's go for a simplified model. Our simple model is based on a coin, the way we can flip heads or tails. And we buy a magic coin in a shop. The question is how many times you need to flip this coin to prove that it's not fair. So, for example, if I flip it 10 times and another person flips it 10 times too, is that the same as 20 flips? Or if I flip this 10 times and multiply it by 10, is that the same as 100 flips? And what if I buy 10 coins and want to know which ones are fair? What would I do then? So let's look at the experiment with magic/weighted coin. Each coin represents a stochastic variable x, and each flip represents an observation xi. The act of performing the coin flip is f, so an observation xi is the same as f of x. We normally assume that a fair coin has an expected value of a half. That means there should be, in the long run, 50% heads and 50% tails. And an unbiased flipper means that each flip is independent of the others. Then you can say the probability of flip 1 of x times the probability of flip 2 should be the same as flipping 1 times flipping 2. And then we should get the same probability. So in the long run, the expected value of the flips is the same as that of the coin. So the expectation of all the flips, the product of all flips, should be the same as the expectation of x as such. So let's transfer this simple model to reality. We have our coin flips, where each flip gives us a small piece of information about the coin and the flipper. More flips provide more information, so we can have random and stochastic variations in coin and flipper. They will cancel out, while systematic variations accumulate. And then we come back to the plot of the target practice I had before. And here you can see that there is systematic and random variations. In the real experiment, each measurement tells us about our sample, naturally, our instrument, and our analysis. So more measurements provide more information about random and stochastic variations in the sample, instrument, analysis, which should cancel out. And normally the analysis should have little or no stochastic variation. Systematic variations, they accumulate. So if we have, for example, a biased analysis, this will clearly show in the results. Now, let's do a more complicated model. Coin flips is actually quite simple and probably difficult to match with another experiment. A very popular dataset that people use for analysis and demonstrations beyond the coin flips is called the IRIS dataset. And if you follow the link on this slide, you will get more information about the dataset. So this dataset covers a number of measurements from different plants and from different species. And when we load it, then we can just call load iris and put it in a data frame, and then we can see a selection of samples, and we can see there are different measurements: sepal length, sepal width, petal length and width. And you see also which plant species that was used for this. Now we can also do a pair plot and show different distributions, and we can already see that the Setosa species actually isolates itself from the other two. And we will see more about this in the following slides. So now we want to compare different groups using the so-called intra-class correlation coefficient. And it's basically looking at how many similar objects within a group are and compared to the similarity between the groups. And in group similarity, we can see how groups are separated. So low group similarity means overlapping histograms, and they are harder to separate for segmentation algorithm, for example. The high group similarity, then the histograms are separated, and it's easy also to segment. Now let's look at the sepals, and here I even have a little illustration what these sepal and petals are. So sepals are the green leaves of the flower bud that covers the flower, and the petals are the beautiful leaves of the flower. And here you can see for the different species how they vary, and you can see that there is quite some overlap in the sizes, so it's not so easy to separate based on the sepal measurement. And the petals, on the other hand, they have a very high group similarity, which means they are much easier to separate from each other. And now let's look further onto this, where we can look at the intra-class correlation coefficient, in short ICC. And the equation you see here is the variance of the averages within of the different classes divided by the variance of the averages plus the average of the variances. This is maybe easier to look at in this slide, where we can also see what it actually means. So if we have 100% ICC equal to 1, that means we have 100% of the variance is between the classes. And that means there is almost no variance within each class. 0 means that their variance is between the classes, and the average values are essentially the same. So the plots down here, we have first, we have averages all overlapping, meaning ICC is equal to 0. If we now have some separations, you can easily see that they are separable in some sense with a threshold. Then we get an ICC already of 0.86. And this increases to when they are completely separated, then you get an ICC of 99. If you would get even closer, then I believe you would have to narrow down the variance within each class, or even separate them even further. So, looking at the intra-class correlation coefficient for our flowers, we can see that for the sepal width, then you have a low group similarity, but and that's also what you can see if you think back on the previous slide. And here on the petal length, then we have an ICC of 96 almost %, and they are much easier to separate. Now, if you want to compare different groups, then we have to first do some kind of hypothesis testing. So first we make a list of assumptions, and from those we can establish a null hypothesis, H0. And usually the null hypothesis is that both groups are the same. Then we calculate the probability of the observation, given that the null hypothesis is true. This requires that you have knowledge about the probability distribution of the data, and the modeling can be really complicated when you build up larger models for the analysis. But in principle, what we are looking for is different outcomes, and here we have the truth in the population and the decision that we make based on it. Then we have reject the null is type 1 error, and retain the null in the type 2 error is the beta. And then we have the correct is 1 minus alpha. And the correct for the type 2 is then the correct power. And if you want to read more about this, I recommend you to look in this Privitera book that tells more on this topic. So let's come back to our coin from the magic shop. Our assumptions were, if we flip and observe coins accurately and independently, the coin, and that the coin is invariant, it should always have the same expected value. So our testing with the null hypothesis is the coin is unbiased. That means we should have an expectation of 0.5. Now we can calculate the likelihood of a given observation from a number of flips. And there we come to the so-called p-value, which is the probability of the observations. So question is, how many observations do we need to be good enough? So for that, we need to take some tricks. And for comparing groups, we use the student's t distribution. Since we usually don't know our distribution very well, we can assume it is Gaussian, and but the Gaussian need more information or more samples, and we usually don't have sufficiently many samples. But according to the law of great numbers, you can at some point always assume you have a Gaussian distribution. Now, with that, we assume we have our distribution of a stochastic variable, which is normal or Gaussian, and the t-distribution provides an estimate for the mean for a given number of observations if you have a low number. And from that, we can then make some likelihood based on our observations. The student t-test uses the student t-distribution, and it provides an easy method to assess the likelihood that we have two observations and that they're coming from the same underlying process, so that our null hypothesis is true. And the test assumes that you have unbiased observation and that they are truly normal distributed. So these are the two base assumptions on the data to make the student t-test valid. So let's go back to testing again on our coin, and this time we do multiple testing. So we had our magic coin, and let's say we found it and trying to publish a paper. Then we have our null hypothesis that the coin is fair. And we heard that the p-value of 0.05, so 5% is good enough. That is true for many observations in biology. If you go to some physics observations, then you need a very much smaller value to be seen as valid. So that means if we get 5 heads, we are good, and we can just go on and publish. And we can see here the probability of increasing number of tosses, that if we take toss 1, we get a head, toss 2, we get a head, and so on. We see that when we come across 5 tosses and all our heads, then we should be good. Now, what happens if we do many experiments? let n friends make 5 tosses. And then we go again for the get 5 heads. Now the equation becomes a little bit more tricky. Now we have first the probability of not getting 5 heads, and that we take to the power of how many friends we have. That turns into this equation. And so let's say we have n friends, then probability of 5 heads is 3.1, like we saw in the previous plot. Now if 10 friends are tossing the coin, then all of a sudden the probability of 5 heads is 27. And so on. If we go up to 80, then we even have 92.1%. This doesn't really make sense, right? No, there is a flaw in this evaluation. And in principle, what the equation does is not what we asked for. The p-value is only meaningful when the experiment matches what we did. We didn't say the chance of getting 5 head ever was less than 5%. We said that if we have exactly 5 observations and all of them are heads, then the likelihood of a fair coin is produced is less than 5%. So there are many methods to correct for this, and mostly it involves scaling the p-value. So the likelihood of a sequence of 5 heads in a row, if you perform 10 flips, is 5 times higher. So then you need to do this correction. And that is maybe not so good. So we really need to do corrections in order to make reliable investigations. So for example, if we want to quantify cell distance to other cells, or oblatedness or distribution of oblatedness, then we need to throw them all through some kind of algorithmic and do our publication. But let's look at what happens. If we have multiple observations from a uniform distribution between plus and minus 1, we make 2 groups. So all these variables, they actually come from the same distribution. And now we want to tell if there is one of these variables that actually deviates despite the fact that they are actually from the same observation. So for that we need to run the t-test, and here is the function for doing this. We have variable I, so that is the one we are currently investigating. That group is equal to 1 or group is equal to 2. And from that we can see that in our little table here that mostly the p-value is more than 5%, but there is one that actually deviates, and that's probably some kind of statistical likelihood that there is one now and then that passes through. And if we do a larger table, of course we do more of these coincidences. But in principle, it should always be more than 5% in the p-value. And now we can do a big experiment. This takes a while to run, but you can anyway test it if you like. And here we have examples of 1 up to 150 variables, and each with 100 observations. And then within each one of these samples, we do even a repeat 50 times. And the plot is now that we find, for one variable, we find only a few significant values. If you go up to 11 variables, then we have more, and then you see there is more or less a linear trend going up here with some spread. And that can also be seen in this different plot, where we use a box plot instead with some outlier markings, and the box telling the median value and the standard deviation. So what we can do now is to apply something called Bonferroni correction, and that's in principle that we take our p-value and divide it by number of tests. And if you look at our previous experiment, then we have this linear trend that it was increasing. But if we divide our tests, or the p number, with the number of tests, you can see that now we are down to the correct significant count, and that makes more sense. And then we have examples when you have made your observations. Mainly all of them are in the same, but one is shifted by 1 standard deviation from the other. And so the average value is shifted by 1 standard deviation from the other ones. And then we check with a different number of variables and run. And then you can see that with our null hypothesis that x equals y, that we usually have mainly, we say that there is no problem at all, but then you can see that there are some that go above the level. And well, these plots show the case of 18 variables up to 87 variables. And you can see that the p-value here changes, decreases for the null hypothesis. And here you can see also that the likelihood of finding a different variable decreases with the number of variables. And from this we can make some conclusions that we often have too little information for perfect analysis. In that case, the student t-test can help, and we need to reject the null hypothesis on a specified confidence level. That is the workflow. And you should, if possible, have repeated measurements in your analysis. And if you do that, you need to correct for the multiple experiments to avoid biases in the conclusion. So that was the end of part 2 of lecture 8. And the next part is about reproducibility and data plotting.